So I'm very happy to introduce today Marieke Grotheer, who is a junior group leader of the Educational Neurolab in Philipps University of Marburg in Germany. So she's been there for 3 years now, but before she did actually a quite long postdoc in the team of Kalanick Spector at Stanford University. So the team is on Vision and Perception Neurolab. And before she did her PhD in the Friedrich Schiller University in the lab of Gina. Okay. So I've been following the work of Marieke for years, and when I realized that she was now in Germany, I was very happy to invite her, and I'm really looking forward to listening to your work, Marieke. So thank you very much for accepting to come here today to Neurospin, and the floor is yours.
Thank you so much for the kind introduction. And actually, I feel really quite honored having been invited here. So you have a very beautiful institute. It's quite impressive. So thank you so much for having me. And maybe just one, can you not hear me?
If you don't hear her, she can use the mic. But if you hear her, for the video, it's fine because it's this mic. So 2 things, if you don't hear her, just tell it and she will use the mic. And the other thing is that Marieke is happy to take questions during the talk. So if you have a burning question, don't hesitate to interrupt her to ask.
Yes, definitely. And please, for the people in the back, if at any point I start getting more quiet and you can't hear me, let me know. And I really like when talks are interactive, so do feel free to interrupt me at any time. Okay, so to get us started, what I would like you to do is I would like you to picture a newborn giraffe. If you like to watch nature documentaries like I do, then you might know that newborn giraffes get up and are up on their feet within minutes after they are born. And actually, they will run shortly after that. In contrast, human infants, a couple minutes after birth, are largely mobile, almost blind, and fully dependent on the care of others. Yet, despite our rather modest start in life, we actually go on to acquire reading and other uniquely human abilities, and to pursue a wide variety of adult careers while also cultivating complex cultures. So on the really zoomed out level, the big picture question that I would like to address in my research is, what is the biological basis of this unique human learning trajectory? And I'm sure there are many different answers to that question, but I believe that one key ingredient to answering this big picture question is myelin. Myelin is a fatty substance that wraps around nerve fibers and thereby insulates them. The more myelin is wrapped around a nerve fiber, the faster signals can be transducted along that nerve. Rodent models have linked increases in myelin to learning, suggesting that myelin is a key component of brain plasticity. So if you prevent myelination in rodent models, they fail to learn certain tasks. At the same time, the overall degree of myelin in the brain increases during development, but this increase is significantly protracted in humans. While in many animal species, developmental myelination is finished by the time the animal is born, in humans, it continues into young adulthood. So this means that the myelin in our brain may be more flexible for a longer period of time. And this potential interaction between developmental and learning-related changes in myelin might underpin the unique human learning trajectory. At the same time, this prolonged flexibility in myelin might also leave us vulnerable, though, and might explain human-specific neurological disorders such as schizophrenia. As such, for me, it's particularly important and interesting to assess how myelin changes when we're learning new things and when we're developing. And in my talk today, I would like to present 3 of my prior studies that were tackling these different questions. So here's a little overview. Overall, I will be talking about 3 different studies. In the first study, we looked at adults and evaluated whether the neurosubstrates or the white matter bundles that are associated with reading and math show different levels of myelination or not. And then in the other 2 studies, I will be looking at how myelin is built up in the typically developing infant brain. Before I jump into the first study, here's just a bird's-eye view of some of the different methods that I will be talking about today, just so that it doesn't get confusing. So across studies, I will be using functional MRI, which measures changes in blood oxygenation to give us an indirect view of neural activity. And I will present all of my functional MRI data in such a inflated cortical representation, where sulci are presented in dark gray and gyri are presented in light gray. In addition to functional MRI data, I will also be talking about diffusion-weighted MRI data. We use the diffusion-weighted data to track the myelin connections between different cortical regions. So you can think of these connections as the wires that connect different cortical regions, allowing them to communicate with each other. I will also talk about relatively novel quantitative MRI measures, and I will talk a lot more about that in detail later. But essentially, these measures allow us to compare tissue properties in the living human brain over time and between individuals. And I will combine these different methods with computational approaches in order to gain a holistic understanding of how the brain, or how myelin in the brain changes with learning and development. So for instance, I will be talking a lot about linear models. So then let's dive right into the first study. Two of the most critical skills we acquire during our lifetime are our abilities to do math, and in the context of this talk with math, I mean simple arithmetics, and to read. In our modern society, these skills are essential for our daily lives. In fact, even a simple task such as ordering a coffee generally requires you to use both of these skills. So even though it's still early in the day, you probably used both of these skills already today. Even though these skills are so important for us, we're not born with the abilities to read and to solve mathematical equations. Rather, this is something that we're trained to do during our childhood education. This training adapts the brain so that it can support these essential skills. Hence, for me, and I know for many of you, math and reading are exciting model systems to probe human brain plasticity. And in this first study that I will be talking about today, we wanted to assess if there's differences in myelination between the math and reading networks in typically performing adults. Before we could answer that question, first we needed to identify the brain regions that are associated with math and reading. And in this particular study, we were really focused on task preferences. So we were not interested specifically which brain regions prefer number stimuli or letter stimuli, but we wanted to keep the visual features of the stimuli constant and really look at task preferences only. So in order to do that, we designed these morph stimuli between numbers and letters that you can see here. And in the actual experiment, the participants were presented with morph stimuli that contained either 80% letter information or 80% number information, while they were performing either a reading, an adding, or a color task. So just to make this a little bit more clear, in the beginning of each trial, subjects were presented with a cue, which told them which task they were supposed to perform. Then they were presented with 4 images, which could either be number-like morphs or letter-like morphs, and then they were presented with an answer screen. In the adding task, they had to simply sum up the stimuli and indicate the correct sum. In the reading task, they had to read out the word and indicate which word they had seen during the trial. So here the correct answer would be bats. And in the color task, they had to memorize the colors they had seen during the trial and indicate which color had come up before. And just because this is usually fun, I'm now going to show you an example trial. So here you were performing the adding task on the letter-like stimuli. And when you're doing this for the first time, it seems to actually maybe be quite hard and counterintuitive. But our participants got really good at this task really quickly. And in case you're wondering, the correct answer here is 24. So in order to identify brain regions that are part of the reading network, we compared neural responses during the different tasks and looked for regions that showed higher responses in the reading task compared to the other 2 tasks. And we found 4 regions of the brain that showed such a consistent preference for reading. These regions were distributed across the entire brain and were more commonly found in the left hemisphere of the brain than the right hemisphere of the brain. Similarly, for the math network, we also found 4 regions that had higher responses to the math task than the reading and the color task. And these regions were interestingly found bilaterally. It's critical to note that this is likely not an all-encompassing list of all brain regions that are involved in math and reading, but these regions were most consistently activated across my participants. So now that we had the functional brain regions that are associated with math and reading, the next step for us was to identify the connections between these brain regions. So for that, we also collected diffusion-weighted imaging from the same participants. And we then performed tactography in order to identify the white matter connections in each participant's brain. And here I'm going to focus specifically on the large range connections, so on the white matter fascicles. I'll talk a little bit more about fascicles later in the talk, but you can think of them as the large-scale highways that connect distinct parts of the brain. So by combining these different types of data, we were able to identify those white matter connections that actually connect to a specific region in the math and reading network. And I will refer to these connections as functionally defined white matter tracks. Given that here we investigate not only a single region, but multiple regions in the math and reading network, we actually had the opportunity to look at how pairs of regions within each network are connected to each other. So for instance, imagine that this is one region in the reading network, for instance, the IFG. We mapped out the functional white matter tracks of this region, and then we mapped out the functional white matter tracks of another region, for instance, the OTS. And then we looked at which white matter connections were actually shared between these two regions. By using the dice coefficient, which is a measure of overlap between two samples, we then quantified how much shared connections there are between the two regions relative to non-shared connections. This then allowed us to generate such a simplified schematic of the math and reading network. So the different acronyms here represent the different regions in each network. And importantly, the width of the lines is scaled by the dice coefficient for that connection. So thicker lines mean that there's a stronger connectivity between two regions. And what was particularly interesting to us was that out of the 13 fascicles that we had tested, only 3 actually played an important role in connecting regions in the math and reading network. And most importantly, the same 3 fascicles were important for reading and math. So these fascicles were the SLF, which is in purple, the arcuate fascicles, which is in red, and the posterior arcuate, which is in orange. Because these 3 fascicles came up in both the math and the reading network, it suggests that they might be a shared neuronal substrate of our abilities to do both of these tasks. However, an open question is whether the entirety of these fascicles are important for both tasks, or whether there might be subsections within these fascicles that are important for only one task or the other. So for instance, imagine that the black lines here are the outer borders of the SLF and the arcuate. One possibility is that fiber tracks associated with reading and math are completely randomly intertwined within this fascicle. This would mean that the entire fascicle is essential for us to perform both of these tasks. However, another possibility is that fiber tracks associated with reading and math remain segregated in the fascicle, so that distinct subsections are important for only one task or the other. And in order to get at that question, the first thing we did was to simply just look at the fiber tracks associated with math and reading side by side. And this is what I'm showing you here in an example subject. So across all our subjects, it very much looked like fiber tracks associated with math and reading remain segregated. So fiber tracks associated with math always stayed superior to those associated with reading. Of course, we didn't just want to look at that, but we also wanted to quantify it across all our participants. And for that, we fit support vector machine algorithms to basically train the algorithm to decide whether a given white matter connection is part of the math network or the reading network based on its location in the brain. And what you can see here is the decoding accuracy across the entire length of the SLF and the entire length of the arcuate. And the chance level here would be 50%. So you can see that this decoding accuracy is quite significantly above chance. So this suggests that there's actually distinct subsections within the SLF and the arcuate that are associated only with math or reading, which for me raised the important question whether we can maybe find differences in myelin between these two different subsections as well. And for that, we also collected quantitative MRI data from the same participants. Quantitative MRI allows you to evaluate the brain tissue structure of individuals in a way that is comparable across subjects, scanning facilities, and time points. It, for instance, gives you measures of T1 relaxation, which is, for me, particularly interesting because T1 relaxation is directly linked to myelin. So 90% of the variance in T1 is driven by myelin. And since myelin changes with learning and development, we wanted to use these new quantitative MRI measures to see whether there's differences in myelin between the math and reading-related subsections of these bundles. So just as a reminder, this is what the SLF looks like, and we find these two subsections that were either associated with math or reading. And now I'm going to plot the quantitative T1 along the length of the SLF, from the most anterior to the most posterior end of the bundle. And what you can see is that those fiber tracks associated with reading show a significantly faster T1 relaxation rate than those associated with math. And we also observed a difference between reading and math related tracks in the arcuate, but this difference was a lot less pronounced. These observed differences in T1 relaxation suggest that the white matter tracks associated with reading are more heavily myelinated than those associated with math in the adult brain. For me, this is particularly interesting because there's good evidence that we actually practice reading more intensely than math during our childhood education. So this might link between practice and white matter myelin and fluency in these different tasks. So in summary, this first study suggests that math and reading involve distributed networks of regions that are specialized on processing only one task or the other. And while the same white matter fascicles are important for interconnecting regions in the math and reading network, distinct subsections within these fascicles are important for only one skill of the other, and they actually show differences in myelination, which highlights the important impact that our childhood education has on our brains. Any questions about the first study? Yes, please.
Yes, so I'm not entirely clear which one is more myelinated than the other.
Yes, so it's going to get even more confusing, but T1 relaxation is negatively related to myelin. So lower numbers mean more myelin, meaning the green one is more heavily myelinated, which is reading. Yeah.
Did you get a chance to study inter-subject variability?
Unfortunately not. We did collect math and reading behavioral questionnaires for these students, but they were all Stanford students and they were all at ceiling in our test, and we also only had 20. So in the end of the day, it just didn't make sense to look at that. But when you have more data with more variance, it would be very cool.
I might question you afterwards to see how you would do it.
Yeah, let's do that.
If you're interested, we'll tell you.
For sure. All right. Cool. So that was the only study that touched on learning and myelin. The other two studies that I will be talking about today will be about the development of myelin at the very onset of our human life, so during human infancy. And you might wonder why I chose to study developmental myelination in infants, given that infants are maybe not the easiest participant population to work with. To answer that question, I would like to show you a little video. In the video, I'm comparing changes in brain volume in the first and second year of human life. Sorry, just gotta get this to start. If you look carefully, you will probably notice that the brain changes much more drastically in the first year of life relative to the second year of life. And in fact, the human brain never changes as rapidly as it does during human infancy. So for me, this makes human infancy a particularly interesting developmental period to study. And in fact, at the end of the video, the brain of a 1-year-old looks much more similar to the brain of a 2-year-old as what we started out with when comparing a 0-month-old to a 1-year-old. So a lot of stuff is happening in the first year of life. And importantly, these volumetric changes are also associated with strictly regulated and important microstructural changes. So here I'm plotting changes in the longitudinal relaxation rate R1, which is also derived from quantitative MRI across the different human ages. So this is what I meant when I said it was going to get more confusing. So R1 is actually 1 over T1. So it's essentially the same measure that I had before, but it's positively related to myelin. So the more R1 there is, the more myelin there is. And you can see that basically the myelination increases across quite a large period of human life, but this increase is most rapid during human infancy. But what is plotted here is actually the average across all of the white matter. So it's not very specific. And what we wanted to get at in the first infant study that I will be talking about now is whether there may be spatial differences in how the brain gets myelinated in early infancy. Or in other words, whether there might be specific regions in the white matter that get myelinated faster than other regions. And then, of course, we also wanted to look at the mechanisms that may explain these differences. So in order to answer that question, we actually collected our own infant data. We collected diffusion-weighted, quantitative, and T2-weighted anatomical images from infants longitudinally. Our infants were scanned longitudinally at 0 months, 3 months, and 6 months of age. And then we used the diffusion-weighted data to identify the white matter fascicles in the infant brain. And we used the quantitative MRI data to measure R1, so to basically get a sense of how myelin is developing in the brains. So we scanned the infants at night during their natural bedtime, and we asked the parents to follow the infants' usual nighttime routine as closely as possible. So the infants were usually swaddled, changed, fed, and then fell asleep, hopefully, in the arms of the parents before we very carefully moved them onto the scanner bed. But whenever, and then we started the measurement, but whenever an infant woke up during the scan had to be stopped and we had to start over. So it was took quite a lot of patience, and it was quite an endeavor to collect this data. But once we had the data, in order to look at the spatial differences across the white matter, we needed to divide the white matter into meaningful units. So in other words, we wanted to identify the white matter bundles within the infant's brain. And here I'm showing you one of these bundles in one of our newborn participants. In this particular case, we defined the bundle manually. We basically just drew our eyes of where the bundle should go through. However, in order to do this across subject and time points, and also in order to make it more reproducible, we wanted to use automatic approaches to identify the bundles across infants. But this is basically where we ran into a problem. So when we used AFQ, which is a very commonly used software to identify white matter bundles that we also used in adults in the last study, it actually failed to identify the white matter bundles in most of our infants. And this is because, as we later found out, because some of the anatomical assumptions that are made in AFQ are just invalid for the tiny infant brain. So before we could even tackle our main question of interest, we actually had to make a new version of AFQ, which we called baby AFQ, that is specific to the infant brain, and which recovered the bundles across our participants. And overall, as you can see in this graph, baby AFQ overlapped much more strongly with the manually identified bundles than AFQ. So basically, it helped us identify the bundles across participants. And baby AFQ is available both for MATLAB and Python, and I'm always happy to help people get started. If you ever want to use it, let me know. So here I'm showing you some of the example bundles that baby AFQ identified and how they changed as our participants grew older. Overall, baby AFQ identifies by now 26 different bundles. At this point in time, it was 24. So we had 24 different bundles in our infants. And for simplicity, so the results are pretty similar across bundles. So for simplicity, for the rest of the talk, I'm only going to show you 2 bundles, which is the corticospinal tract, which is in orange, and the anterior thalamic radiation, which is in blue. So once we had the bundles, we could finally look at how R1 changes along the length of the bundle. And the first thing you might note is that R1 does indeed change as the infants get older. So the dotted line are the infants at their first scan, the dashed lines is at the 3 months time point, and the solid line is at the 6 months time point. So R1 does go up, but it actually changes not in the same way in different white matter locations. So the increase in R1 was different between different bundles, and it even varied along the length of a different given bundle. So for the rest of the study, we tried to figure out what are the mechanisms that drive these spatial differences in R1 development. And the first thing we found is that the growth of R1 in any given, oh, was there a question? Go for it.
May I ask just what is the standard error or the standard deviation for on the model?
Yeah, so basically there's this like shading, right, which is the standard error across the infants at that age. Does that answer the question?
Yes, meaning that it's probably a bit larger if you were talking the dots of any of the charts.
Right. We did look at the individual subject data, and it does go up in each subject, but there is a decent amount of individual variance. Yes.
Thanks.
Mm-hmm. All right, so the first mechanism basically that we found is that the growth of R1 in any given location in the white matter depends on how much R1 there was present there at birth. So the solid line here is the data that we collected at the 0 months time point. So it's the data that was collected shortly after the infant was born. The dashed line, on the other hand, is the R1 growth rate. So it's a slope of the R1 increase. And you can see that these two things are negatively related. So in areas of the brain where there was relatively little myelin at birth, R1 increases more rapidly than in locations where there was more myelin at birth. Or in other words, the white matter is basically playing catch up, which might mean that it's trying to homogenize differences in myelin that existed at birth. And the second thing we found is that R1 increases over the first 6 months of life follows spatial gradients. So that is what you can see in this figure, which I totally admit is a little bit hard to pass. So the different dots here are different locations across our white matter bundles. And in the first figure, they are just colored based on what bundle they belong to. So that's relatively not so important. In the second figure, however, they're colored by the R1 slope. So based on how quickly R1 grows in this particular location in the white matter. And basically what we found is that R1 increases along spatial gradients. So it increases more quickly from the inferior to the superior and from the anterior to the posterior axis of the brain. So overall, this study found that myelin grows rapidly in the first 6 months of life, but that this growth in myelin is not uniform across the brain. The spatial temporal differences in myelin growth that we found can be explained, at least in part, by how much myelin was present at birth and by spatial gradients of development. Yes.
I have many questions, but maybe just keep on if you want. First, this one on the spatial gradients, maybe counterintuitive to what I would have expected for the frontal thing. So you say it's progressing from anterior to posterior?
Yes, meaning that in the posterior it's faster. So once it gets closer to early visual cortex and so on, it myelinates more rapidly than the higher level cognitive functions in the front.
But you also say that the slope was higher in the regions where the myelination is lower at birth.
Right.
So this would suggest that in the frontal, you have more myelin at birth than in the...
True. Yeah, that's a good point. These two effects are sort of orthogonal to each other. So what we did to get at that is that we fit linear models where we entered information about R1 at birth and about the spatial location. And basically knowing both of these things together best describes how R1 changes in a given location. So it's both of those, but the spatial gradients was definitely the stronger effect. So the R1 at birth, I thought it's my favorite effect because I think it's quite cool, but it was much smaller compared to the spatial gradients.
And my other question on a different thing is that, okay, in this protocol, you had both this quantitative one mapping and the diffusion. So you use the diffusion acquisition only for the tractography, but you could imagine also deriving some of the DTI parameters.
Like FA and MD.
Yeah.
We did that actually. It's in the supplements.
Okay.
What we found there is that MD very closely matched what we found in R1. So maybe it also is a reasonably good measure for myelin in infancy. But FA was completely different. It showed completely different effects, which I think is fun because not too long ago, people often equated FA with myelin, and at least in infancy, that doesn't seem to work so well.
So that's an interesting thing, I guess, for the ones only being able to acquire diffusion data,
right,
to know this.
Yes, I would recommend MD. It actually correlates quite highly. I have a figure on that later also.
I'm curious about the variability that you mentioned before across participants. Could you say on which of these measures, like is it the myelination at birth or the trajectories, or which one is particularly?
Across individuals, basically. So we only had 10 individuals, so it's a very tiny study, and we didn't look at that specifically. So we did notice that across people on average, the spatial gradients is a much larger, more robust effect. But I gotta admit, we didn't look at the individual subject level because it was such a small sample. But I have a bigger sample coming up in the next study. Cool. All right. So this leads me to the last study I wanted to talk about today, where we compare differences in myelin growth in utero and ex utero. So in the moment that we're born, our environment and experiences drastically change. We move from the dark, sheltered, warm environment of the womb to the outside world. And we suddenly have to breathe for ourselves. We have to eat for the first time. We are bombarded with all the sensory input that we didn't get before. And basically, what we wanted to know in this study is what is the impact of all of that on the myelin in the infant's brain. And this last study was published last year, so it's still relatively new. So to get at that, we basically contrasted these three different hypotheses. The first one is the boring one, which basically says being born doesn't change white matter development. The slope just continues as it did in utero. The second hypothesis is based on the idea of experience-dependent myelination and suggests that all of this extra input that you're getting after you're born promotes myelin, so that myelin growth is faster ex utero compared to in utero. And the third hypothesis is exactly the opposite. It suggests that the sheltered environment of the womb that has like a constant supply of nutrition and oxygen and so on, maybe that's really good for myelin development. So maybe myelination actually slows down after the infants are born. And to get at this question, this time we took a slightly easier route. So we did not collect our own infant data. Rather, we used the data that is made publicly available by the Human Connectome project. And we used the second data release of the project, which made data from about more than 300 infants publicly available. So everybody can download this data and play with it. And we divided this data into two samples. What I will call the cross-sectional sample had 300 infants that were born either full-term or preterm, and they were scanned shortly after birth. In addition, we also derived a longitudinal sample, which only contained preterm infants. So all these infants were born before 37 weeks of gestation were completed, and they were actually scanned twice. So they were scanned once after their preterm birth, and then once again, once they reached an age of 37 weeks, or in other words, once they reached a full-term age. And then we used some held-out data to basically generate an age-matched control group, which were infants that were born full-term, but they were scanned at the same age as the second scan from the preterm infants. So basically, this contained two samples of infants that were scanned at the same time, but one group had more time developing in utero than the other group. And since we were particularly interested in myelin development in the study, we again wanted to use a measure that is close to myelin. However, unfortunately, the Developmental Human Connectome project does not actually contain any quantitative MRI data. So now we had to use something else. They do provide you with the T1/T2 ratio maps, which they themselves call myelin maps. So we decided that we would use those. But it should be noted that whether or not these maps are a good measure for myelin is definitely still debated in the field. So what we did is we used some of our old data from the previous infant study to basically see how well does this ratio actually correlate with our quantitative measures of myelin. And you can see that at least for infants, it does correlate quite well. So R1 does correlate, or T1/T2 does correlate stronger with R1 than MD and FA, whereas MD also still correlates much better than FA. So all of that basically just made us a little bit more confident in using this measure for this particular study. And then we again used baby AFQ in order to identify the white matter bundles in all of the different infants. And here you can see a few examples of infants that were scanned at different ages. And again, for simplicity, I'm only going to show you data from the first 3 bundles, which is the anti-thalamic radiation, which is in blue, the corticospinal tract in orange, and the cingulum cingulate, which is in green. But the results were really very consistent across all of the different bundles. Okay, so we started by evaluating the cross-sectional sample and asked whether there's any development in T1/T2 that is dependent on at what age the infant was scanned. And importantly, whenever I say age in the context of this study, I mean gestational age, so age relative to conception. And in order to get at that, we first pretty arbitrarily divided the data into 3 different age groups. We had a 33 to 37 weeks age group, which are all preterm infants, and that's a solid line, and we compared that to 37 to 41 weeks and 41 to 45 weeks. And as you can see, the T1/T2 ratio does increase depending on the age of the groups. So there does seem to be some development happening here. However, importantly, at the time of measurement, the infants had a certain amount of time developing in utero, and they already had a certain amount of time developing ex utero. So this does in itself not yet tell us what is the impact of birth on white matter myelin development. So to get at that, we fit linear models in which we added the infant's birth age and the amount of time that they spent between birth and scan as separate factors. So this allowed us to assess what is the impact of birth itself on in utero and ex utero development in each of our infants. And the first thing we noticed when we did that is that the model that had information about when the infant was born largely outperformed a model that only had information about when the infant was scanned. So there does seem to be an influence of birth on T1/T2. And in the graphic here, you can also see why that is the case. So here I'm comparing the slope of T1/T2 development between in utero development, which is the dashed line, and ex utero development. And you can see that the in utero development is actually much faster. So the white matter myelin develops much faster in utero as opposed to ex utero. And this accelerated or deaccelerated ex utero development is quite interesting in the context of preterm birth, because infants that are born prematurely, they don't have enough, or they don't have as much time to develop in utero compared to their full-term peers. So in the next step, we basically focused on the longitudinal sample, and we wanted to see whether there's differences in white matter T1/T2 between these 3 different groups that we had. And this is what I'm showing you here. So the solid line are the preterm infants at their preterm scan, so right after they were born. The dashed line are the same infants once they reach the gestational age of 37 weeks. So same infants, but scanned again at a later point. And the dotted line is the age-matched control group. So these are full-term infants that were scanned at the same age as the dotted line, as the dashed line, basically. And what you can see is that the preterm infants at their second scan, they're somewhat in between their first scan and their full-term control group. So they're fallen a little bit behind in their T1 over T2 development, which might be linked to the fact that they had less time to develop in utero compared to their peers. So in the last step, we wanted to explicitly test this. So we wanted to explicitly test whether knowing when an infant was born can explain their delayed white matter development that we see in the preterm infants. And for that, basically, we used the linear models that we had fit on the cross-sectional sample to try and predict the data that we had seen at the preterm infant second scan. And this is what I'm showing you here. So the colored line is the same data as you see before. It's the data from the preterm infants at their second scan, where they have fallen somewhat behind their full-term peers. The dashed gray line at the top is the model prediction that you get from a model that only knows the scan age of the participant. And you can see that that largely overestimates where the infants should be. Now, importantly, the dotted black line is the model that has also information about when the infant was born. And you can see that this model that separates in utero and ex utero development actually predicts the data at the preterm infant second scan quite well. So that suggests that the relative amount of time that preterm infants have to develop in utero and ex utero actually explains quite a lot of the variance or the delay that you can see in their white matter development later in life. So overall, our data showed that ex utero and in utero T1 over T2 change showed slower ex utero than in utero T1 over T2 change, which is consistent with the third hypothesis. So it suggests that the sheltered environment of the womb, which is constant supply of nutrition, may indeed be beneficial for white matter development, and that it sort of slows down right after birth. And overall, the study suggests that being born has quite a fundamental impact on the white matter myelin development in our brains. Questions about that study?
Yeah, maybe one. Did the group that you used for training the model included the subject that on which you tested?
No, that was independent. So the training group did include preterm infants, but it's different individuals altogether. All right. So in conclusion, these studies I showed you today, I hope contributed to our understanding of how myelin changes when we're learning new things and when we're developing. In my future work, I plan to evaluate how these two factors interact in order to understand how learning changes when we grow up, which in my hope is that it could contribute to our understanding of the unique human learning trajectory. Thank you for your attention.